So the very quick tour from eye to the brain, to the extent that you can start to think about how this backward masking, binocular rivalry, flash suppression, and continuous flash suppression might be happening, and also form your hypothesis with related to this. Okay. So the first thing is about the eye. If you are interested in consciousness, you might find it boring, but bear with me. So this is the lens of the eye, and then at the back of your eye, there's a layer called the retina. Okay, and the retina has this dip here. Do you guys know what this means? It's actually the opposite. It's the best part of the vision called the fovea. But that's actually, a bit strange. Why this, dip is the best? Do you have any idea? Who said that? Or maybe the other people is better. You want to say?
No.
I got it. I was thinking maybe because it's more bent there, so maybe light just reflects better and you're able to capture it more. I don't know.
Okay.
Isn't it where the cones and the
Rods.
Yeah, like you find them most in those areas, like the
Right.
cones.
Right. But so that's a factually, it's correct. But why this bended part is the best in terms of vision? That's the question. Do you have any idea or mechanistic explanation?
No, I was going to go for the rods and cones.
Right.
Like high definition. Is it rods or is it?
That's good as a sort of what it is.
Yeah. But why is that?
But why this part is the best?
Like, so more light can hit the cones or the rods.
I see. That's an interesting idea. So that kind of is an optimally structured to obtain the light. Yeah, in a sense, it's correct. Do you have any opinion?
Something to do with neural convergence, because the outside areas wouldn't activate properly if it's all spread out. I don't know.
I see. So some kind of like concentration of the neurons or resource.
Linear inhibition. I mean,
Inhibition
lateral inhibition.
with respect to the surround.
Yeah, so if it hits one spot, it can't focus on the outside spots, which means the eye is required to have a centralized spot for information. I don't know.
I see. Okay. Yeah, it's actually quite difficult to think about why this invented part is the best part of the vision. And one thing that is actually hinted in this figure is this part. Fatima, do you want to speak up? Do you have an all good? So Fatima says that the more cones are placed there. Yeah, so it is true that more cones are placed in this phobia location. But the question I want you to think is that why this bented part, this structure gives you better vision. And the hint is here. You usually miss it if you don't pay attention. This part. So what this part means is that if you place the electrode, that's the experimental kind of condition, right? And then put the needle into here, then you find some kind of neurons. It's called the retinal ganglion cells. And that spikes like this way. When something hits around the so-called receptive field of these neurons. Okay. Do you guys know what this is? Then, this side. So this is the retinal ganglion cells, neurons. And you already mentioned something about retinal structure, which already contains this answer here. What are these things that looks like some kind of shape here? Do you have any idea? Who? Did you say something? No? No idea? No idea? Exactly. Yeah, that's right. So these are the rods and cones. This part. That is not weird, no? Who thinks it's weird? Maybe you said first.
No, I said, why is it?
Normal, you think? Okay. Does anybody think it's a bit abnormal or doesn't make sense?
I'm just confused. I don't know if it's the diagram. Like, why does the diagram only highlight those cones and rods? Like, do they serve a more special purpose there?
Yeah, I understand why you can be confused, but this is just sort of simplistic explanation of the eyes, but it contains important information. What it means is that when the light comes in from here, eye, right? Then it gets first passing this neuron layer, and then arrive at cones and retina, cones and rods. What's the alternative architecture? If you are the God, and if you want to make a better human, what would you do? Yeah, maybe. Lina.
Switch the cells so that it first passes the rods and cones and then goes via the ganglion cells to the brain.
Exactly, right? Isn't that what you expect? Yeah. Right? It's weird. And because we don't have that, because this guy actually, interferes with the processing, they put all of these, ganglion cells outside. So somebody actually said that, putting a resource outside, that's actually correct half, correct? And then this thin layer is devoid of, ganglion cells and any other stuff. And so it directly, more or less directly goes to the cones and rods. And that's why it's thinner and why we can see better. But in the first place, why don't we now have the cones and rods in the, surface, right? Does a okay, Caitlin asks, okay, sorry, I missed something. First, ion channels are on the cones and rods. And then second question is it is part of the retina, and the retina is made up of rods and cones. Is that right? Yes, that's correct, Fatima. And does the fovea only consist of photoreceptors? It's not only, even if it's thin, there are lots of things that's still going on. So if we don't have anything, in between, that's much, much better. But we don't have that architecture. It's weird, right? And this architecture is discovered, I think I said to you guys in the lecture 1 or 2 or 3, that it was the Descartes who did the anatomy of the sheep eye and then discovered this. And then at that time, I also said something wrong. Sorry. I think I said something wrong, that octopus actually sees the world in a not inverted way, but that's wrong. But one thing that is correct. So that's sorry, that's one thing. But one thing that is still correct is that the octopus is inverted in other way. So light comes here, right, from the top. And then in the case of humans and other cones and rods are here. So it's all diffracted and interfered up to here. Look at this octopus. They have these cones and rods just beneath. Yes, Kisaj.
So in the fovea, our site is basically like the octopus, where the photoreceptors are on top while the networks are at the bottom.
Close, but not quite. And I don't know exactly how much you don't have anything above, but I think you still have something above. So there is the fovea is our fovea is still not really optimum. But if we have this kind of architecture, that's much better. That's super obvious, right? And octopus has it. We don't have it. Why? Nobody knows. Okay, so in fovea, there's no ganglion cells. I think that's correct, but I don't know. So you guys need to probably do the Googling or ask ChatGPT whether there's no retinal ganglion cells in the fovea or not. And let us know if you find the answer. But my point here was that octopus has this inverted structure, which was so interesting that I spent so much time this week, and that resulted in some problem in the structure of this lecture. So I can talk a little bit about the octopus as a digression. Maybe can I talk about it a bit, because this is so interesting, right? So octopus eye has this lens function. So it projects the image onto the inverted way, like us. So that's my mistake, right? Okay, but they have this very strange kind of lens shape. It's square shape, not like us, circular, right? That's one thing. And then evolutionary point of view wise, there's some kind of branch of the according to evolutionary history, humans are the part of this vertebrates, right? Fish, and then from here, branch into another things like mammals and also birds. I think birds are also here, I think, and the reptiles, right? And amphibians. Really long time ago, we had this branch. And then here the branch into echinoderms, like a starfish, that's the one, that happened after. That contains something similar to us, okay? And then this, very different guys here includes this, like shrimp and then worms and the octopus and also flatworms and planaria and also so on. And then there, almost none of them has this lens-like shape of the eyes. And this mollusks one, the octopus kind, has something similar, but everything else doesn't have anything similar to our eye. And this is how it looks like. So human eyes look beautiful circular shape right here, and then the mollusk part, like that contains octopus, squids, and also other shells, shell-like fish. And then this is a planaria. Planaria has this cup eye. It's very weird. The octopus has this camera eye, but other ones like pinhole or mirror or compound eye, like insects. So it's totally unclear how these eyes of the octopus actually emerged. At the same time, it also makes sense that it has a completely different kind of architecture. Maybe it's even more ideal than humans. But interestingly, recent neuroscience and also behavioral science actually revealed lots of interesting things about octopus. Octopus is probably very, very intelligent. I don't know whether you were born at the time, but there was some octopus who bet accurately to the winner of the football World Cup, consequently, like 10 times or something, right? That's a fake news, but they can see motion, shape, and remember, and then make some kind of inference logically. It's very, very wise, and also it seems to communicate with other octopus, but they live really short time, like only 2 to 3 years. They die, so it becomes like it's like us human develop really quickly and then die, and then do all these complicated things, which is very unnatural if you think about all the things that are told about why human takes time to develop intelligent brain and so on. So octopus is super interesting. And this one I wanted to show. That's the anecdotal kind of video, octopus. And I knew about this kind of thing before, but this year, like June, in Nature, my friend, some later in Okinawa Institute, published this paper quantifying exactly that, yes, octopus indeed sleep like us, first of all, scientifically he proved. And then also during this, dreaming, he found that some pattern of the coloration happens like during the, wake, quantified in a very, very computationally, complicated way. But it's very convincing, and that's why it went to the Nature paper, right? And this, the right side of this other minds is also very engaging and interesting philosophical, monologue that I recommend if you're interested in this kind of thing. But the point of all this is that the closest case of encountering a conscious alien creature, probably, we can think of. And in many ways, it's very different, but it's very intelligent. And also something very interesting from the consciousness research point of view is that because their skin is so rich in terms of reports, unlike our behavior, right? They can actually report what they are experiencing through their body online in a precise way. It's very, there's this, huge potential in terms of this reportability of their experience. And they can actually match their statistics of what they are seeing with their color. We can't describe it, right? How do I report, my seeing of this blue? I just can't say just, blue or similar to that blue. But an octopus can just, express it. So that's a very interesting point of this octopus. All right. Any thoughts on octopus consciousness? Do you have? Is the camouflage behavior automatic or objective? All right. So do you want to elaborate on this? Whose opinion? If not, then I'll just supply some more context then. So the camouflage behavior, I don't know the details, but it seems like if you watch that video, that the person who raised this in octopus says that when the octopus sees the crab or something that they want to eat, then they change the color and then to hide into the sea. So I think it's probably intentional. It's not like a automatic thing. Does it count as a language? Interesting. Whose question? Or do you want to say anything about this? Well, what is actually the condition of language? That's the question, probably. What? Okay. Suddenly, lots of things appear. Is there some kind of delay here? Maybe?
I don't know.
I see. Okay. All right. Then also another thing is that is there they're intelligent enough? Can they report anything? So I think yeah. So if you hide some objects and then you need to press a button or open the bar to get there with a several logical induction, they can do it. So it's intelligent enough. And then other things, fascinating. Thank you from Octopus. And then other things of particular interest must have evolved long ago. Yeah, that's true. But this evolution-wise, the other mollusks do not have the same, eyes, right? So they evolved their camera eye and also all these visual behavior relatively recently. So octopus itself is probably, has evolved a long time ago, but some of the function resulted relatively quick recently. Okay, is there any particular thing you wanted to say? I just feel like this word cloud is interesting. Yep.
What I was trying to write was, before it separated all my words, was it's interesting if octopuses do have consciousness, a consciousness at the same perhaps level as humans, then on our pursuit of understanding our own consciousness and doing our experiments with humans, we can try and also kind of replicate those on octopuses to see if there are any similar behaviors or neuronal activity we notice there, or even if there are distinct, activities we notice with the same experiment we design to test for consciousness in humans.
Right. So one thing I learned through this Peter Godfrey Smith, who wrote this book, is that according to him, when we are under the pain, usually we tend it, right?
Yeah.
Unconsciously. Like if this is painful, you just, rub very gently. Yeah, itching.
Yeah.
Right. However, octopus doesn't seem to do that. I don't know whether this is really true or not, but something like that is fundamentally related to the behavior and also quality of the pain, and whether that's necessarily the case that we behavior and pain quality is going together.
I was just wondering, could there also be a difference between it being underwater and the senses just being a little different?
Yeah. You mean pain?
Pain, like it just feels a little different, or
Yeah. Maybe, but I don't know. The dolphins and also whales might tend their scalp potentially. Because it was raised as a potentially very different kind of, consciousness or quality from us. All right, now back to the track. Coming back to the human world. And then, so there are, I talked about the retinal ganglion cells, right? I skipped a lot of complex, processing that is already known and also interesting on its own within the eye. And they came to the spiking neurons called retinal ganglion cells in the eye. And they tend to like some kind of spot in one particular area of the visual field. So if your ganglion cells respond to somewhere here, while I'm looking at this corner, then if I flash the spot of light here, then that goes like here. Yeah. But if I show anything else, like a different color of the stimulus, then it goes silent. And then if you show the preferred, anti-preferred stimulus here, like black against white, and then this one is white against black, right? Then it goes off even below the baseline. That's the sort of the typical behavior of this ganglion cells or any neurons, for that matter. And then the opposite of this off-center cells are also, expected. And you can imagine something like this for the red-green contrast and the blue-yellow contrast, and that's the sort of a cone-related, ganglion cells. But it's already, cones and rods are differently, mixed in terms of timing and space to generate this. So it's already complicated kind of processing going on. And then now further skip into the brain. So this back of the brain, back of the eye, retinal ganglion cells pass the information to so-called thalamus, center of the brain. It has been always difficult to explain, but now with the thing here, it's easier. So thalamus is in this C part. Okay, maybe you can pass. So this is pretty much in the dead center of this, brain, and it's hard to access from anywhere. And then that's where this thalamus is. And then from thalamus, there is an axon going into visual cortex here. That's a primary visual cortex. But there are at least 10 different pathways from eyes to the brain. And I'm also showing only one of them, which is most dominant for probably conscious visual experience. And then if you already notice, there's already something going on here in terms of coloring. Do you understand what this color of the red and the blue means in this figure? Did you say yes? So what is it?
It's the left side and the right side.
Yeah.
The two different pathways.
Right. So
Yeah.
the right side of the vision goes to?
The opposite side.
Yeah, left hemisphere, right? Now, this already comes to part of the answer of what you experienced in the beginning of the lecture. Do you know what it is? This figure and also the answer from Jack kind of hints at some of the answer or is a mechanism as to what you experienced in the beginning of the lecture. Which of the phenomena that is related to this architecture? You experience backward masking, binocular rivalry, flash suppression, and then continuous flash suppression. So which one and why? Do you have any idea?
Like the left side of the brain kind of took priority in a way.
I see. So when you see two things going to the left hemisphere take the priority. That's actually that's not this figures implication, but indeed in Monash University there's a scientist that claims this hypothesis actually. So it's not a bad idea really, but you might have already kind of guessed, but this is something to do with the binocular rivalry or continuous flash suppression or CFS, right? Because when you see two different things, especially around here, with your left eye or right eye, then that goes into left brain and then processed as one thing. Okay, so within left hemisphere and within right hemisphere, right eye and the left eye competition is already once resolved first. And then after that, as you said, there is some kind of interpretation like thing happening in the brain, and that's something that some people claim that it is the left hemisphere that is determining which one you are seeing. But at this point, important thing is that within primary visual cortex, at each point in your left visual field or right visual field, input from right eye and the left eye is converging. And if it's not the same, then that's the reason why you see this binocular rivalry. Make sense? Any questions so far? Okay. All right. So then a little bit more of the anatomical explanation. This is a different format of the brain, but it is based on the real anatomical flattening. So once you get the dead monkey's brain, and if you are very skilled, then you can tease apart the brain and then flatten it like this way. And also connection from B1 to LGN is preserved, and then this one is the retina in the back of the brain. And this is the first step for the anatomist to, understand how the brain areas are connected. Like medical student, if you are lucky, you do this kind of things. And then in terms of the property of the neuronal response, as I said, at the level of retina, you only respond to the spot of the light. But at the V1, if you come to the cortex, then you start to see response property to become complex. And here, do you see what's the complexity introduced here? It's still simple, but it's much more complicated than the retinal ganglion cells. From this figure, do you see? So what it shows is that, let's say, if I'm fixating here, right, as a monkey, and then you show this grating around here, and then you are recording the neurons that respond around that part. Okay, can you imagine that? So I'm now sticking an electrode like this to monkey, and then if that neuron responds to this area, then if I show the stimulus like here, then it shows fires, becomes active. And it used to be the case that if it's in a retinal cells that respond to the same location, no matter what orientation I present, if it hits on that same location, it goes like whatever. But here, if you see it, it responds only in here, right? Most strongly. And what it means is that when I see at the fixation and then I show this in this particular orientation and the particular direction here, but it doesn't show activity in the other direction. So that's the complexity introduced in this V1. Yes. Sure.
Does it mean like only direction of movement could make the neuron firing, like the V1 neurons?
This V1 neuron, yes. So other V1 neuron may be, that's a really good question, and may be sensitive only to the orientation. And may like only very slow motion or very fast motion. All sorts of the different kind of property can be found. Remember how many neurons are there in the brain? 86 billion, and 70% is in the cerebellum. So just like 10 billion or something like that is across the cortex. And visual cortex is in fact very small, but it's still big portion here. And the V1 is about this one, this yellow thing here, that's V1. Okay, this yellow strip in the back of the brain. I don't know if you can see it, so I'll just show it over there. Can I take that? Yeah, V1 is very small. It's in here, this number 3 in this thing. Okay. That's V1. It's very close to this cerebellum. So in terms of location and the motion direction and orientation and also size and so on, each different neuron has a different preference. That's in V1. Okay. Is there any question? Yeah.
Like it's a little bit just for what's the C bits for?
C.
Like the brain part region C.
Thalamus.
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 For
That's an excellent question. What do you think is the thalamus is doing? Well, then, in other words, if I ask, if you were to record the neurons from thalamus, would it be more similar to retinal neurons or V1 neurons? Because retina sends input into the thalamus, and the thalamus is an input to V1, right? And the V1 already responds to the line of the particular direction, and then retina only responds to the spot. So there must be something happening in the thalamus, right? Yeah. Who thinks that the thalamus already responds to the line? Who thinks it's only responding to the dot? No idea? So you're okay. I mean, yeah, it's an unfair kind of question. I mean, the fact is that the thalamus doesn't do much. So it responds only to the spots, pretty much. Almost like a retina. However, I mean, I'll just hold it. There is something weird going on in also, thalamus that is very unique to thalamus, but not in the retina. If you have any idea, that would be great. But maybe let's hold it later. No further question about this? Okay. Then, so in general, the higher in the hierarchy of the visual area. So it goes from thalamus to the V1 and then V2, V3, V4, and so on. That's the hierarchy. And then around here, there's a visual motion area, and there's a visual face area, and there's a text area, and things like that. It's very, very complicated. It becomes very complicated, and nobody understands why or how this kind of thing happens. Another thing is that the bigger the size of the receptive field becomes, and also complicated, as I said, right? And another architectural property is that the similar neurons tend to be found in neighborhood. So if you find a neuron that responds to this location, for me, when I'm fixating here, then the next neuron is also likely to be responding to somewhere here. And also, if the previous neuron likes this direction, then the next neuron is likely to respond this direction or something like that. And also, if that one responds to the right eye input, but not to the left input, then again, the neighborhood neuron is likely to respond to the same eye input. So similar neurons tend to be located within the visual cortex in the neighborhood. And that's very important when you think about the integrated information theory, but that's another story. Probably I need to explain it in week 10 or 11. So the figure here shows that the V1, V2, V3, V5 goes from the back of the brain to going this way. And then V1 is, as I said, it's embedded in here. So it's difficult to see in this figure, but that's how this back of the brain is dedicated for the visual areas. And as I said, the number of the Vx increases, it becomes bigger in size and also more complicated. And then maybe I'll just skip here. For example, by one example of the complexity is that V2 starts to respond to the illusory line, not the actual line. So V1 neurons would respond to this kind of real line. But a V2 starts to respond something like this. If start and end is there implied, but not to this. Because here there's some kind of gap in here. It's not that V2 neurons are thinking or expecting that, there's gap, so I shouldn't fire or anything. There's some very mechanistic thing going on there. And that slight difference here just automatically does that. And then here, it's an implied, gap right here. That also, activates this neuron really strongly. So as I said, B1, B2, B3, B4, as it becomes higher, it's more complicated. And in AI, if you are interested, there's a technique called deep learning. And then also, from the first layer to go to the higher layer, the response of the unit becomes more and more complicated. So we kind of start to understand why this complication can, emerge from different kind of complexity of the connections and so on. And so learning loop. All right. And then MT responds to visual motion. Okay, Caitlin asks a question. Does this illusory line relate to the Gestalt principle of visual cognitive interpretation? That's an excellent question. What do you think? Potentially, yeah. And who said yes? Yeah.
Is it like have some similar because they're like non-voluntary? It's just like neuron instinctively doing this, not through learning, but it's just like it could respond in this way, just naturally. Yeah, like because they do not detect the line with gap.
Probably yes. But whether this is actually directly related to Gestalt principle or not, that's also difficult to say. Because you are not this neuron, right? And the Gestalt psychology is talking about how you experience these things to be done. You know what I'm saying? So here, you now start to think, start to come into this, very enigmatic thing about neuroscientific finding and your phenomenology. You yourself experience that as a connected or not. And then there is one neuron in the brain that correlates with your experience like that. But what does it mean? So that's a difficult question, but a very good question. Okay. And then this is motion and so on. And I'll just skip this. And then before the break, I think I want you to, now I think you have all these, ingredients to think about potentially how this backward masking can happen. And I'll just remind you that in backward masking, I show some stimulus very briefly, right? And followed by that, I just show lots of lines. And then if the delay between the two is very short, you don't see the first stimulus, right? You convince that by yourself. And then sometimes you see it, sometimes you don't at the threshold. But if you look at the anatomical connection of this brain, and then going from I to LGN to V1, V2, V4, IT, and then prefrontal cortex, and then motor cortex to make a response that I saw it. There's a delay of 20 milliseconds, 30 seconds, 30 milliseconds, 40, and so on. By the time it's here, it's roughly like 100 milliseconds or so. And then to get to the prefrontal cortex, it's 130 milliseconds. And then you can make a response within 250 milliseconds, okay? in the case of monkey. In human, it's slightly slower. So information flows, right? When you see it, there's something going on. And then after that, you see the backward masker, that's also processed. But you see only the second one, masking one, not the first one. Why is it? Why do you think it could be the case? So this is a model that the philosopher came up with, right? You show, so the x-axis is the time, you show some kind of dots, very, very free onto your retina. And then that causes a neuronal activity. Like, retinal ganglion cell or LGN or, B1. And then after some threshold, you start to see this. It makes sense, right? Probably. There's at least some kind of delay. But if that's the case, why is it that backward masking has to happen? And yeah, this is one attention hypothesis, but maybe I'll just stop here. And then I want you to think about this. I think I made it in this case, not the word cloud. You can give a little bit more elaborated response. Do you get what my question?
Is it like how what we learned about neural activity could explain the mask phenomenology?
Right. Or not? If it's difficult to write, maybe you can just say that's also fine. Yeah.
I was thinking, since all the processes take like individually to go from like V1 to V2 takes 40 to 60 milliseconds, and it all adds up to before going to the spinal cord and then to the finger muscle. Maybe the mask is so fast that it overrides the pathway, and instead of seeing the stimulus before, the new stimulus overrides it, and you see the mask instead.
Excellent. So that's one really sort of the predominant hypothesis. Now, so what's your name again? David? So David's idea is that it may be the case that targets takes time to process, but second mask is actually faster, and it catches up and then overrides. That's what you're saying, right? Is there any idea to support or refute that kind of idea, or do you have any idea?
I thought maybe letter has more meaning than the lines, and that takes more time to process then.
I see. Yeah, that's also another good point, in fact. So Lina's idea is that because the letter is more complicated kind of stimulus than lines, it takes more time to process and also emerge into consciousness. So that's why this meaningless thing can catch up and erase it. Is that? Any other ideas? Or so maybe there's something. Detailed objects take longer to pass along this chain of neurons compared to something that is less. Is this similar to what Lina said? Yeah, okay, good. Iconic memory has an incredibly limited storage capacity, which would be automatically overwritten by new visual information. That seems like a very knowledgeable kind of answer. Is that from you guys or from TA? No? You guys? Okay. Huh? Yeah. Yeah. What's your name, by the way? Oscar.
Iconic memory is the split-second visual information that we take in through our eyes, and it, I think, only lasts 8 seconds usually. And I think there's it has an incredibly limited capacity according to Sci 3051, which I took last semester. I don't know.
Yeah. Okay, so that's a sort of a visual flash memory.
So flash memory you can take in when presented with about 9 letters, you can take in about 3 letters at a time.
Right.
So I think therefore, if it's only clouded by new visual information, it would be automatically deleted.
I see.
As there's not enough time for it to even enter into your, working memory.
I see. So what Oscar is saying is that even though you kind of took this L information or K information, because further information of these lines actually overflow the capacity of this iconic memory, you only see the later later part of the things. Okay. But how does it actually, do you think, relate to this, brain-based kind of explanation about, iconic memory?
At least what we learned in our last unit was that we just don't know how it relates to the brain. There's not enough information. So
that's the thing that we need to think about, right? Okay. Yep. So any other answers? Let's see. New response, does meaning influence our perception speed? That's very interesting idea. And what do you think? What's your hypothesis? Do you think that the meaningful stimulus takes more time to see? Okay. Then who thinks a meaningless stimulus takes more time to see? Okay. Maybe do you wanna somebody who haven't said much? Maybe you.
When there's an internet phenomenon where you can have words where the center letters are jumbled up, but the ones on the ends are in the right place, and yet you can still very easily read the words despite the letters being in different orders. Therefore, you're actually processing the meaning of the words first rather than what you're actually seeing. Therefore, it makes me believe that interpreting meaning can be faster than actually perceiving.
I see. Okay. So based on the internet phenomena, you are saying that maybe the meaningful one is easier to perceive?
I remember in 1 week when studying perception and cognitive psychology, there's this research that was conducted. So a person was asked to pay attention to the words that is being said to their left ear, but and they have to report back what they hear from their left ear. So they have a headphones on, and I believe, I could be wrong about the context, but like on the right ear, it's like 1 ant, like ant, A-U-N-T, 1 ant and 8, and for this one is like my 9 Jackie. So when you think about meaning, it makes more sense to think of like my Aunt Jackie. So what they hear on the left one is not my 9 Jackie, but my Aunt Jackie. But Aunt was like on this one. So it's like meaning precedes how people like, but they admit that they were paying attention to the left ear instead. So I thought like meaning is easier to perceive.
I see. So in other words, you're saying that the meaning is grouped before attention acts, so that even if you try to attend to the right ear, because the grouping acts at the level of the meaning, you go from left to right, or left kind of ear switching, right? That's what you're saying. Yeah, that implies, again, sort of the depth of the processing at the level of meaning may be much faster than the meaningless thing. That's your idea. So both Daniel's and Jagger's ideas is kind of, meaning is faster to be processed. Is there forward masking? That's another good comment. Whose comment? Yeah. So this is not to ask your opinion, but having in general, the magnitude is smaller. But if you reverse the order, like in line, lots of lines, then letter, you also don't see the letter. But in this case, you need to make the letter much fainter, and also lines more stronger and things like that. But that is challenging to Oscar's idea, right? because the order of the things would say that, first thing would occupy the iconic memory storage, and the later things should come later, but it cannot come in that situation. Think about it, all right? So, but I don't have a phenomenology here, so I can't really give you these things. Explained by recency bias. Okay, that's a good idea. Whose idea is this? Yep. So what's the recency bias? Maybe you can.
Just like the kind of tendency to emphasize that what was most recently shown to us, like the most recent stimulus.
I see.
Because there's also a primacy bias, except that's based on the idea that memory has been consolidated between that time. But since this was so quick, whatever was most recent would take priority.
Right. Yeah, so I guess you might have heard of this primacy and also recency bias thing, but this is kind of contradictory term, right? This is the thing that I don't like about psychological, explanation. But the recency bias means that later thing is more emphasized, and that's a backward masking. And then the primacy bias is that fast thing bias. That's a forward masking. But what determines which one operates? That's unclear according to this theory, right? So you need to have some kind of guidance or rule to say which one operates at which time, which case.
I mean, the idea behind primacy bias is that memory has been consolidated, and that usually happens within 30 minutes.
Right.
But because this was so quick, there's not enough time for memory to consolidate. So it makes sense that primacy bias doesn't work here.
I see. So primary things related to vision is more recency bias based. That's your case idea, right? Okay, that's very good. And then other things, sensory memory being overwritten. I think that's what we discussed in terms of iconic memory. Letter has more meaning than lines. Okay, so that's also related to meaning based explanation. I think that's pretty much good explanation, and thank you for the hypothesis. Keep active. Okay, for the rest of the, so just to wrap up this backward masking part, and I'll leave this binocular rivalry part for your sort of the exercise to think about. Maybe you can write it in an essay. In the case of the looking at what happens in the brain, if you have a capacity to record from everywhere, this is what happens roughly. At each moment of time, not like strict hierarchical, B1, then B2, B3, B4 kind of thing, but many things actually becomes active at the same time. That's how the brain works actually. Okay, so this is very complicated, but if you look at this, before 40 milliseconds, B1 and MT is called B5, but that gets already active. All right, and then around 65 milliseconds, that part of this, very complicated prefrontal cortex is even already active, and so on. And so this almost simultaneous, but very fast response is something to keep in mind. And then this guy called Victor Lame, it's not exactly the same as recency, but the kind of related idea of the recurrency idea is the one that he proposed. So at the time when the stimulus is presented, the first stimulus is presented here, 0 to 40 milliseconds, there already this part is active, right? And then or after that, mask starts to come in. And then when the mask starts to come in, this V1 area starts to be already active about mask, right? If you don't have the mask, then it forms some kind of a reverberation from this later activated area, IT, and that's the sort of meaning area that you guys were talking about. And meaning and V1 gets kind of communicating and so on, and then that establishment of this recurrency loop is the one that the Lame suggested as a origin of consciousness. And backward masking disrupts this communication. The first path goes to the already meaning part, and the meaning part tries to kind of confirm whether that's actually true, but that's already not there. So you don't see it. That's the idea. Make sense? More or less? Okay. And then the alternatively, Stan Deheng, who proposed this global neural workspace theory, said something similar, actually. And there isn't a difference between the Lame's recurrency hypothesis, but it's kind of similar. So according to Stan Deheng, what's quite important is to think about the strength of mask. Strength in terms of the meaning or contrast or duration or whatever. That goes from really low to high. And as you saw, if the duration between the target and the mask is long, then you see it, right? So you can see the duration can be a parameter of the changing the stimulus strength. And if you look at this V1 area, then regardless of the stimulus, the masking stimulus, stellar strength, it gets activated like this initially, like 100 milliseconds. You see a lot of activity initially. But then after 200 milliseconds, depending on whether the mask was strong or weak, the weak case is conscious, and then that's here. And then strong case is this, blue and green, and that's here. You see the difference. And the reason is because of this, recurrency-like thing, feedback, according to Stan Dehaen's case. And Dehaen says that it is a prefrontal cortex here, and also parietal areas that shows this bifurcation here, conscious case versus unconscious case, that can enhance this, activity at a later time through this feedback loop or not. And then also there is some kind of, high visual area, like, ideas as well. But Dehaen thinks that high visual areas without prefrontal is not sufficient. So that's the difference between the Lame, who thinks that, prefrontal is probably not necessary here, his vision doesn't have this part, right? Versus Stan Dehaen. And that's the kind of the discordant, theoretical explanation, but roughly speaking, two things are suggesting something similar. Okay. Yeah, and I'll just stop here, and then I'll give you some further references and so on in the slides. But if you already have some things to write in the essay, you don't need to check that optional reading. Okay. That's it for today. Thank you.